Okay, hello everyone. My name is Vilas Minartovich, and over the summer I worked with Dr. Shafiki, and the culmination of this was this project where I studied a gene of my choice, which I chose the Hox gene. Um, but we worked to learn more about PCR, how it works, and what it can do. So my presentation is Hox genes and homeobox sequence primer design with Primer Blast, a recap of the Horizon Quest internship 2025. So introduction to Hox genes. Hox genes are a family of transcription factor crucial for body plan development. They're highly conserved across different species, and they encode proteins with DNA binding domain known as the homeobox. And mutations in Hox genes can lead to developmental disorders and diseases such as cancer. So what are primers? Primers are short synthetic DNA sequences, approximately 18 to 25 nucleotides in length, and they provide a starting point for DNA polymerase in PCR, which is polymerase chain reaction. They bind to a complementary sequence on the DNA template, and this allows for the amplification of specific DNA segments. The key properties of this are the GC, or the guanine and cytosine content, melting temperature known as TM, and specificity. How primers work in PCR. The first step is denaturation, where DNA strands separate at high temperatures. The second step is annealing, where primers bind to their complementary sequence. And the last step is extension, where DNA polymerase extends primers, synthesizing new DNA. And as the cycle repeats, it exponentially amplifies the amount of DNA fragments. So, primer blast. NCBI's NCBI Primer Blast tool designs PCR primers with built-in specificity checks. This ensures primers are unique to the target sequence, and it prevents amplification of unintended regions known as paralogs or pseudogenes. And it provides the TM, GC content, and predicted PCR product size. Hox gene primer design example. So I chose the Hox D13 gene, and the forward primer results and reverse primer results are as follows. The PCR product length was 172 base pairs, and both primers had a GC content of 55% with a TM of 60 degrees Celsius. Um, and this primer, Hox gene primer design example, it designed outside the conserved homeobox region for specificity and was verified using Primer Blast against the genome. Okay. Why specificity is critical. Hox genes share nearly identical sequences in the homeobox, and non-specific primers may amplify multiple Hox paralogs. This leads to unorganized gel results with multiple bands, so ensuring specificity guarantees accurate results for gene expression studies. Strategies for primer specificity we learned were for my specific gene was avoiding the conserved 180 base pair homeobox region, verifying uniqueness with NCBI Primer Blast, and matching primers TM values closely, and lastly, use hot start DNA polymerase to reduce non-specific binding. PCR optimization. Annealing temperature optimization is crucial, so starting 10 degrees Celsius above primer TM, decreasing it gradually, and then you can run temperature gradients plus or minus 5 Celsius around the TM. And so the ideal result would be a single sharp band in gel electrophoresis, and you can confirm product identity by sequencing. So this was a screenshot of the results on the NCBI Primer Blast website, and I chose Primer 1, and this was using the Hox D13, as I mentioned earlier, in mRNA sequencing. Here's another screenshot of the specific results of Primer 1. And then we used in silico PCR as well to simulate how it would go in real life with the expected products. And then here's a screenshot of those results. So why I chose Hox? I chose to make a primer for a Hox gene because these genes control bodily layout during development and are highly conserved, making them great for studying gene regulation. I was interested in how small genetic changes can lead to big differences in form and function, and what we learned over the summer. Hox genes play a fundamental role in bodily plan regulation. Primers are essential tools for amplifying specific Hox gene regions. Primer Blast ensures primer specificity and efficiency. Careful design avoids amplification of conserved regions. Successful primer design enables accurate genetic studies. And we also learned how to use and navigate NCBI Primer Blast and the use of UCSC Genome Browser for in silico PCR, as well as learning all the fundamentals about PCR and how it works and what it is. Thank you so much for your attention, and if you have any questions, feel free to ask.
Thank you, Vilas. It was very interesting. Anybody has any question for Vilas? No questions? So in that case, Vilas, do you want to also share your experience? Like, what did you enjoy the most during your summer internship program, and what do you look forward to How
Yeah.
you're planning to use this knowledge in future?
So I think the part that I enjoyed the most was being able to research a specific gene of interest, and being able to, learn all the nuanced aspects of it and how it can work in real life, because these are the real websites that you use when you're actually trying to do this process. So I thought that was super cool to be able to experience. And I'm looking forward to utilizing this throughout the rest of my scientific engagement and career. If I ever need to do this in real life, I'll know how to do it. So that's a plus. But I just thought it was a really cool experience overall, and I learned a lot. So it was really great.
Yeah, and I can give you a pro tip that bioinformaticians are always in like high demand. So this can be a very good career choice if you want to pursue. So that's you
Yeah,
learn very good fundamentals for that. Congratulations.
thank you.